The recording now. Okay, hello everyone. I'm Stephanie Black, the Director of Continuing Education for the Ontario Shelter Medicine Association. Thank you for joining us today. This meeting is possible due to the generous sponsorship of Ogena Solutions. Ogena Solutions was formed from the company Anavac Corporations, which was created in Canada in 2005. Ogena Solutions is dedicated to providing complete, effective, environmentally friendly biosecurity solutions, equipment, and protocols, as well as groundbreaking patented animal bathing systems. Besides being the exclusive master distributor for Burox Technologies' accelerated hydrogen peroxide disinfectant products in the Canadian animal health sector, Ogena's own equipment, products, and matching usage protocols are respected as being the best and most efficient products available in animal health today. Ogena Solutions, sorry, I'm just gonna I have a lot of people that have their cameras on, and I'm just gonna turn your cameras off because it's gonna show up in the recording. So if everyone can please make sure your camera is turned off, so you're not going to end up showing up in the recording, that would be appreciated. Otherwise, everyone will see your smiling face there. So if you can please turn off your cameras and mute your microphones, that would be appreciated for the sake of the recording. So Ogena currently operates in the animal health sector, including farms, shelters, veterinary facilities, pet boarding, and daycare applications. Biosecurity, green technologies, and leading-edge bathing systems are central to the company's product focus. So thank you to Ogena Solutions. A few housekeeping items before we get started. Dr. Gordon will be giving a presentation followed by a question period. Questions can be asked in the chat window, and we will ask them on your behalf at the end of the lecture. Please keep your microphone muted throughout the presentation to avoid feedback and background noise. Dr. Emilia Gordon is the Senior Manager, Animal Health for the BC SPCA. Her primary role is to provide animal health support, training, and oversight to all sheltering branches province-wide. Her areas of focus include infectious disease prevention and management, facility housing design, and sanitation. Dr. Gordon also works with veterinary students, participates in community partnerships and outreach, conducts shelter medicine research, and volunteers on the Canadian Veterinary Medical Association Society of BC Veterinarians Chapter Animal Welfare Committee. So please join me in welcoming Dr. Emilia Gordon.
Thank you so much, Stephanie. And I want to thank the Ontario Shelter Medicine Association and Dr. Linda Jacobson and, of course, Ogena Solutions for making this session possible. We are excited to share not just about the discovery of the novel Fechavirus, but also to talk a bit about the outbreak, because I think there's lots of info and learnings in there for shelters, and I know that we learned a lot during this, this outbreak. So as Stephanie said, the presentation will be followed by a question period, and the presentation essentially follows sort of like a case report format, but near the end I will switch gears and talk a little bit about kind of the research side of what ended up happening. Definitely we welcome questions at the end, but also during the presentation, especially if there is something that's not clear or not clear on the slides, it's fine to interrupt me via the chat window, and then Stephanie will just interrupt me. So let's get started. So I've included, Dr. Jacobson came up with this catchy title, and I've also included all of the collaborators on the publication that eventually happened as a result of this outbreak. And I will talk more about everyone's role in the acknowledgements. But I'm just going to briefly mention the BC SPCA because I know we have participants on here from all over and not everyone is necessarily local. So I work for the BC SPCA and we operate 36 sheltering branches as well as other programs throughout the province of British Columbia and Canada. Our mission is to protect and enhance the quality of life for domestic, farm, and wild animals in BC. And in 2019, we rescued almost a little over 21,000 animals in our facilities. So that's our shelter facilities as well as our wildlife rehabilitation center. And the total number of animals that we helped through all of our programming is almost 42,000. We also conduct cruelty investigations for the province of British Columbia under the Provincial Prevention of Cruelty to Animals Act. And so we conducted over 8,000 cruelty investigations last year, and we have other programs that help both animals and people. And one of our programs is our provincial shelter medicine program, which is just me and Mandy Idle, our registered veterinary technician. She's one of the co-authors on the paper. That's a picture of her, and she's also on the call today. And I just wanted to briefly introduce her because without her work on both the outbreak, but also the data curation and the research piece, none of this would have come together. And I'm not even sure the outbreak ever would have ended. So I wanted to make sure to credit her as well. And as you see photos of cats, you may wonder, like, why are they blurry, or why are they not, professional stock photos? And the reason for that is that I thought it was really important to make all of the photos in this presentation actual cats who are part of this case. You'll see the cats and you'll see their names and you'll see them as they were photographed for our adoption site. And I think that for those of us who do a lot of population level work, it is really important for us to always keep in mind that these animals are individuals with individual needs, that when they come into our shelters, our first goal should be to keep them healthy and provide for their welfare. And that even though you'll hear me say numbers about what happened in this case, we should always bring it back to these individual little animals. So this is Grayson, and he is also on the last slide, so you'll see him again. So just getting into the clinical piece of this case. When we see vomiting and diarrhea in cats, we often aren't too concerned, especially if it's a single incident. Anyone who's owned or worked with cats knows that these are pretty common clinical presentations. Sometimes they're also part of more chronic illnesses that are related to other things. But in shelter medicine, the time we get worried is when we see GI illness that seems to be spreading between animals, or is just occurring above the baseline for that facility, even without a clear pattern of spread. And when we see this, there are kind of three categories that the possible causes fall into. The most common one, and the one that we think about most as veterinarians, is infectious reasons. So viruses, bacteria, parasites, anything that could cause inflammation or irritation in the GI tract. And many of these are zoonotic. So it is really important for us to keep that kind of One Health aspect in mind. If we are dealing with an outbreak, then by definition, we have transmission within a facility or building. We need to make sure that we're also in the back of our mind thinking, are dogs getting sick? Are people getting sick? And if so, that also helps us to narrow down what may be causing it. The other things that are maybe less recognized, but I've seen two different kind of outbreaks that I investigated that turned out to be diet related, is diet. So something with food or treats that maybe they're contaminated, maybe they're spoiled, or a toxin. So we don't like to think about it, but either an accidental or even deliberate poisoning of confined animals under our care is possible. And so we don't want to get tunnel vision when we start to see these cases. We want to make sure that we investigate all possibilities and essentially kind of at the same time. The other thing I'll mention, and you guys don't need me to mention this because you've been living in this world for the last 3 months, is that early recognition and early mitigation for outbreaks is really important. I won't go into that further because I think everyone understands why. And through this case, we'll talk about how we kind of did our best and maybe what some of our learnings were about things we could have done better. And when we investigate outbreaks, we kind of do multiple things simultaneously. One thing that we really want to understand is what is causing the outbreak. And that's important because that helps us design all of our measures, all of our control measures, all of our testing measures. That also helps us design our population management measures like quarantine and isolation periods and where those animals are placed, and also measures like stopping animal movement or stopping intake, those types of things. It's also really important during outbreaks to document everything, and especially in an unknown outbreak like this one was throughout the whole time we were working on it, just to document like who is sick, where are they, what day did they get sick, what day did they get better, those types of things, and to communicate with all affected stakeholders because these types of outbreaks reach far beyond the individual pages of the animals that are affected. So this case started on a rainy, cold weekend in November of 2018. This was actually a while ago. And on that day, I was at home, it was Saturday, and I was checking my laptop for lab results because they all come in on Saturday for some reason through our provincial shelter medicine program. So what I saw was 3 negative fecal tests from our shelter in Vancouver from these 3 cats. And this may not on the surface appear to be any cause for alarm. However, our protocols are such that we don't run fecals on all cats. We don't have the resources to do that. So we essentially only run fecals when cats are sick and there's no other apparent cause. And usually they're kittens with diarrhea and we find stuff in the fecals. So the first red flag was how many fecals there were from one facility, and the second was that they were all negative. So I reached out to our manager, Jody Dunlop of our Vancouver facility, and I said, Jody, are you noticing any GI disease that's spreading between cats in your facility? And she said that over the last week and a half, there had been this kind of sporadic GI illness, which again hadn't reached the level of suspicion to be an outbreak, but that now on this day, on this weekend, we had 8 out of 12 cats who were sick and they had vomiting and diarrhea. So right away, we knew that there was something strange. That's definitely above the baseline for one of our facilities and cause for alarm. And we stopped all animal movement in the shelter that day. We implemented level 3 PPE, which for us is gowns, caps, shoe covers, and gloves, and everything except the shoe covers are changed in between each animal within the room. So that's the level that we would use for something like ringworm or something like leukopenia. We actually based our levels of PPE on the excellent protocols from the Toronto Humane Society. So thank you, Dr. Jacobson. And so we started doing that right away. We also started tracking all the cases on a spreadsheet to track who was where and who was experiencing what clinical signs. And we also looked at what diet they were on and what oral medications they'd been given, just trying to look for any patterns. We started a fecal library of samples that were diarrhea. And on Sunday, we decided to close the cat areas to the public because we had even more cases. And on that day, we also pulled all the food. We put samples of the food in the refrigerator from the most commonly fed types of food. We're on the Hills shelter feeding program, so we were feeding Hills Science Diet to most cats, but not all cats. So not all cats were on the same food. So that was one clue that it probably wasn't dietary. That was an early clue. And we washed out all of those bins and just changed everything out completely just in case it was something related. And then on Monday, when everyone was back in the office, we initiated a full outbreak investigation for this case. And what we discovered was that 2 kittens had come on our through our transfer program from our shelter in Cornell in the middle of November, that they had been vomiting on the truck, coming off the truck, and placed in the shelter using our normal kind of like level 1 biosecurity protocols, which are robust enough to prevent the transmission of most diseases. And what happened was that when we reached out to the Cornell manager, Colby O'Flynn, we learned that, again, sporadically over the last week and a half or so, she also had seen 9 cats and kittens with vomiting and diarrhea in her shelter. So at this point, we've got over 20 cats and kittens who are sick. And we traced it back to the index case, and that's patient 0, or kind of the first case. And this was 3 kittens who had come in from a rural area near Williams Lake, and they had developed vomiting and diarrhea within a day or 2 of entry to the shelter. And there were no cases prior to that in any of our shelters that we could trace to this case. So we believe this was the first case, and it was these 3 adorable, fluffy, little innocent-looking kittens. At that point, we realized that our standard fecal testing, which looks for parasites, basically, we probably needed to expand it. We were really worried about something bacterial or viral. And so what we did was we asked for some special funding, and we submitted multi-pathogen diarrhea panels on 6 cats from the shelter. We typically try to test 30 to 50% of an affected population, and these of the cats that were currently sick, that ended up being about 6 cats. And so we submitted those PCR panels, and what those are panels that look for the genetic material from various different known pathogens that can be run by commercial labs. And the nice thing about them is that they can be run, you can get a lot of information from a single sample. They are a bit more expensive because of what they entail. So they're not a first line that we would normally run for a cat with 1 episode of diarrhea. We also stopped cat intake because we were realizing that we couldn't flow cats through and move them out responsibly. We didn't know what was going on. And we updated the senior management and communication team because we started to realize that this was going to affect our shelters and that we were going to have to at least partially close a couple of shelters. And sadly, on that Tuesday, we lost our first cat, and she was euthanized. And she was euthanized because she had ongoing diarrhea that wasn't really responding to supportive care. And also she was anemic and she had tested positive for feline leukemia on a screening test. At that point, our threshold for euthanasia was not super high because we knew that there was value in necropsy and understanding what was going on. Unfortunately, her necropsy was inconclusive, and also her confirmatory testing for feline leukemia was negative. So this is not the topic of this webinar, but another reminder that making euthanasia decisions based on single screening tests for feline leukemia is not ideal. And so we, unfortunately, this was the first cat that we euthanized, and there was one other. And we had hoped to get some more information from her, but we unfortunately didn't. During the next week or so, we had kind of a series of disappointments, which was the test that we ran. So all of those PCR panels, things that we had kind of added on to the PCR panels, like rotavirus we tested for that's not in the normal panels, they all came back negative, and Merlin's necropsy came back with some non-specific inflammation, but nothing that could explain the outbreak. And she was euthanized in kind of the latter part of her illness, where she just had this persistent diarrhea, but it wasn't during the acute phase of illness. So the pathologist wasn't able to find even anything that was consistent with a type of pathogen, much less a specific pathogen. At that point, we did get on the phone and called Dr. Kate Hurley, a shelter medicine specialist at UC Davis, and she connected me with Dr. Patty Pesavento, who's a pathologist at UC Davis. They were involved in discovering virulent systemic calicivirus together, and talking to them was really helpful. It helped us understand that we needed to gather more samples, that our infection control measures were along the right lines. They gave us some other avenues for investigation, and they recommended some other specialized tests that we could do through IDEX. So we went ahead and did those, and we went ahead and started saving samples for Dr. Pesavento as well. And what you can see here is this pattern of what was happening with us at the time. And so what that was that shelter 1 is the Cornell shelter. So that shelter had cat 181, 182, and 183. Those are the last 3 digits of their animal ID numbers. Those were patient 0. And you can see to the right of those patients that other animals in the Cornell shelter became sick. The ones that are kind of bracketed together in groups are ones where we understood that illness spread from one group to another, but we couldn't pinpoint the exact transmission pathway between cats. And we also, one of the interesting things about this is that all of the transmission initially was indirect. So none of these cats were housed together, none of them had any direct contact or played together. The dotted lines, the dotted arrows indicate indirect transmission between animals. So you can see that cat 614 and 853, Rocky and Pablo, were animals that were then moved to shelter 2. So that line indicates that they were moved between shelters. And the first cat in shelter 2, Vancouver, who got sick, Jelly Belly, cat 688, that illness went kind of it wasn't something that we were super alarmed about, but then what happened was this next group below 688, where there was this large explosion of illness, and that was where we had the number of cats that were sick that enabled us to detect the outbreak. At that point, we implemented control measures, and there were a few more cases off to the right that were traced to contaminated things like an intake room or a vehicle that was used for multiple cats in the same day. So we did have an explanation for every time our infection control measures failed. And I'll talk about shelter 3 now. So in early December, kind of one of our worst fears was realized, which was that we had this satellite adoption center that the Vancouver shelter works with, and they house all cats communally in a really nice space, and it's associated with a private business. And so they reported that they had cats there that were sick, and rather than trying to segregate that population and isolate them within that facility, we just brought all of those cats back to Vancouver, emptied out that facility, deep cleaned it, introduced new cats. Those new cats got sick, emptied out the facility again, deep cleaned it, and then used it for rabbit events for 3 weeks. It's a pop-up adoption facility for a local rabbit rescue, emptied, deep cleaned again. What deep cleaned means is that everything was sprayed with accelerated hydrogen peroxide at the appropriate dilution and contact time. Everything that couldn't be or was washed with heat, like washing and drying in a washing machine, Everything that couldn't be laundered was thrown away. So this was like a full clean. And in early January, we thought, okay, that has to be sufficient for whatever is going on here. Let's start sending cats there again. And we didn't have any cases in Vancouver, but by mid-January, cats started getting sick there again. And it was initially like 1 cat or 2 cats, and then it was almost all the cats except for 1. So at that point, Mandy did go to that satellite adoption center, and she helped them deep clean, and she identified a number of fomites that were not in the cat housing area. So they had not been discarded when the facility was deep cleaned, but they had been touched by people who had touched cats and then potentially touched by people again. And efforts had been made to clean that area, but there were some mostly like loose objects that had been missed. And unfortunately, in the middle of December, that was the 2nd cat that we euthanized. Millie was an older cat who also had other concurrent medical problems. And it was the same thing. She got acutely sick, had supportive care, and then just developed diarrhea that wasn't resolving. And so she was also euthanized and submitted for necropsy. And again, over that month, we did all the additional specialized testing through IDEX. UC Davis sequenced what was in our samples, and nobody found anything. And Millie's necropsy wasn't informative, and we still didn't know what we were dealing with. But despite that, we knew that we wanted to bring this outbreak to an end. So we did carry on with our measures. We had regular team meetings with these 2 shelters. We continued to not take in cats and slowly tried to empty the shelters by adopting recovered cats out with waivers. And we didn't know what we were dealing with. So initially we held them, but we realized that we didn't have cats that were relapsing, and we didn't have any transmission that we could trace to recovered cats as we gathered more of like the epidemiologic information over time. So we knew that we had to move the cats out, and that's what we did. Then we closed both of those shelters and we cleaned them for 2 days each. And we resumed normal operations in the beginning of January. So at this point, we had this theory, and this is the outbreak transmission diagram again. The satellite adoption center, after Mandy's visit, had no further cases. So they did introduce new cats and had no further cases ever since then. And you can see on the bottom here is a satellite adoption center, shelter 3. And you can see that it was initially a couple of cats that were sick. These red lines are clean breaks that had failed. Typically there were only 2 or 3 cats there at the time, but only affected cats are shown in this diagram. And then on the far right-hand side, you can kind of see this group of cats where there were 9 out of 10 cats who got sick. And these cats were all brought back to Vancouver because Vancouver at that time had really stepped up. They had the isolation space and the comfort dealing with recovering and recovered cats. And we had really stringent biosecurity measures in place to bring cats back into Vancouver. But unfortunately, despite all of those measures, 1 day after this group of cats came back into Vancouver, we had 1 cat in Vancouver who started throwing up, and for 1 day, and that was cat 849. So we believe that case was, if it was related to the same outbreak, that it was related to the cats returning. And what this pattern kind of led us to was a theory that we were dealing with some kind of highly durable pathogen that could remain viable for a long time, and that had probably a pretty low infectious dose, because all of the transmission, except for at the satellite adoption facility, which was communal, all of the transmission was indirect, which is pretty weird. And it was indirect despite extensive cleaning and disinfection and biosecurity measures that really suffice to manage any other type of disease that we ever see in our shelters. So we developed the theory that we were dealing with some kind of non-enveloped enteric virus that was very durable and that created illness that was similar to human norovirus. So there's literature about human norovirus that has a short incubation period, and what we did was we continued to kind of tweak our control measures by looking at the human literature. So again, like kind of a One Health approach, but in the opposite direction, where we're actually just trying to learn what we can do to manage what we assumed was some kind of tiny non-enveloped durable enteric virus that we couldn't identify with any type of commercial test or existing kind of test that we could do in an academic setting. And with that, we were able to terminate the outbreak. This is an epidemic curve. You guys know what these are from watching the news and reading the newspaper over the past 3 months. And basically, we have the number of new cases and like along the x-axis along the y-axis, and the number of days along the x-axis, and this goes all the way from November to January. You can see that the green cases are the first facility where it appeared, so that's our Cornell facility. The blue are Vancouver, which was the second facility, and that big spike is what really enabled us to recognize this as an outbreak and start to take measures. Within a few days after we implemented measures, we were able to really reduce the number of new cases. They became sporadic, and we were able to identify clear breakdowns, which always involved fomites in all of the places where our infection control measures had failed. And so, but it was disconcerting when we had this cluster at the end there. But what that enabled us to do was, at that point, we had given up completely on getting a diagnosis, and we, except for possibly through research analysis. And so what we did there was, anytime there was any diarrhea or vomiting, we collected it and we froze it right away. And those samples ended up being the highest quality samples that we had. So in summary, the outbreak itself clinically, there were 43 cats that met the case definition in the 3 shelters that were included in the study. There were actually 74 cats that met the case definition overall, because there was a 4th facility that was affected but was not part of the analysis because we didn't have any samples or clinical or epidemic data. It was also along the transfer route, and we just use the same measures there as we use everywhere else, and they eventually were able to terminate the outbreak there too. So a number of cats, we had lots of cats with more than 1 clinical sign. That's why the number does not add up to 43. You can see that the vast majority of cats did have both diarrhea and vomiting. Vomiting is likely a bit underreported because we had a lot of vomit that we couldn't assign to a particular cat in that last wave of illness in the communal housing. And about 2/3 of these cats required an outside veterinary visit. So they had to go to a full service veterinary hospital and receive fluids, medications, that sort of thing. So while mortality was really low, these cats were very sick, and the incubation period was 1 to 7 days. So we knew that the minimum incubation period was really short based on the data that we had around animal movement and exposure. We couldn't always pinpoint exact exposure because these cats were housed individually most of the time. But we knew that in a few cases it was as little as 1 to 2 days. And then with most cats just kind of having the acute vomiting followed quickly by diarrhea, either recovering spontaneously or getting supportive care within that kind of 4 to 5 day range. And anybody that was an outlier, we had this one cat that was sick for 19 days. Any outliers were like it was all diarrhea. So we didn't have anybody that was vomiting for more than a few days from this illness. And as I mentioned, all the cats were recovered. They were all adopted except for those 2 that we had to euthanize. And there was no transmission that was traced to recovered cats, and there were no clinical relapses. So like I mentioned, we had some high quality samples from the last round of illness. And we still felt that these samples held some promise. So even though nothing had been discovered so far, we had a chance meeting with Dr. Roxanne Chan from IDEX Laboratories. She was here in Vancouver for something else, for a conference, and we met with her. And over lunch, we managed to persuade her to take our frozen samples from the last round of the illness and have another look at them. And those samples ended up at UCSF, and we didn't hear anything for about 6 months. And during that time, of course, the outbreak was officially over and we had no further illness, and we had sort of thought that we would never learn anything else from this. But in late September of 2019, we heard from researchers at UCSF, Dr. Yanpeng Li and Dr. Eric Delwart, that they had found a new feline virus in some of the samples. So what they did was they took 5 of the highest quality fecal samples, including one from Lucy here, and they pooled them together. And then they basically put that sample through viral metagenomics, which is a molecular approach where they amplify all the DNA and all the RNA in the sample, and then they compare all of those sequences to known viral genomes in an international database. And any similarities, so anything that looks like part of this sequence is a known viral family, they can investigate further, and then they can go back and sequence those viral genomes from the original sample. So that's what they did. And through that, this is another one of those 5 cats. I just love his face. He looks so unimpressed. Through that, they discovered a new parvovirus in the parvovirus, or the Chifamo parvovirus genus. It was named Fechavirus for feline parvovirus. The next most recently discovered member of this genus was canine parvovirus, so that name kind of made sense. And then they went back and they tested all of the 5 cats individually, and found that all of the 5 cats from those high quality samples, all of the fecal samples were positive for this new virus. And they also tested all 4 of the vomit samples, and all of those were positive for the new virus. And they then went back, so then we got excited after all of these months of wondering what might have happened and thinking that we had kind of lost hope of ever discovering anything. We went back to IDEXX and we went back to Dr. Pesavento at UC Davis and we said, hey guys, do you have any of the frozen samples that we previously submitted? Because we had sent these kind of samples in a patchwork to different places. Many of them had been frozen and thawed, or they weren't from the acute phase of illness. They were from, a few days into it when we kind of got our act together and realized we should collect samples. But at the same time, we felt like these samples would be valuable to check. So we managed to scrounge up 17 samples from our outbreak, and that represented 19 cats because there was 1 litter of 3 that was pooled together. So in total, 8 of the 17 fecal samples and all 4 of the 4 vomit samples were positive for the new Fechavirus. And they did find multiple other viruses. You can read about the patterns in the paper. As one would expect from a virome of cats in an animal shelter, it was pretty cool what they found. But none of the other viruses had a pattern or were the type of virus that would be consistent with potentially causing this outbreak. And I think it's really important to note the limitations here, that we cannot say that Fechavirus caused this outbreak either. All we can say is that it was associated with the outbreak and it was discovered in the highest quality samples. And we didn't discover another potential cause, but certainly we need more research on this virus to really understand what it can and can't do. This is the family tree of part of the parvovirus genus. You can see the Chifamo parvovirus, that bracketed section. These are the Chifamo parvoviruses that have been discovered in diverse species of animals, with the blue dots representing the 2 sequences from the 2 individual cats, where the whole viral sequence was determined from this study. And you can see there at the very, very bottom, canine parvovirus. So the other carnivore parvoviruses that we know about that affect domestic canids and wildlife, they are related, but not too closely to this new virus. And so just kind of wrapping up here, some of the things that we learned from this, these are the 2 kittens who brought whatever this outbreak was from Cornell to Vancouver, Rothy and Pablo. And one of the things that this reminded us of was how the conditions in shelters really predisposed to disease transmission and to disease emergence. Things like having multiple animals in one facility, and particularly if housing is communal so that animals are contacting each other's body surfaces and potentially are exposed to a higher infectious dose of whatever it might be. It could really be any dermatologic or respiratory or gastrointestinal pathogen. The transfer of animals between facilities, which is a life-saving, it's a life-saving program to be able to move animals from parts of BC that have very few people, but lots of animal overpopulation, to areas like Vancouver, which have tons of people and little animal overpopulation. This enables us not only to save these animals' lives, but also to create some capacity in the source shelters to do more community-oriented work, with the ultimate goal of, we always talk about how ultimately we don't want to need to do this, but it's a short-term kind of measure for us. But the reality is that transfer programs do amplify the transmission of pathogens, and it's something to be aware of. For example, in this case, it would have just remained in the Cornell shelter had these animals not been moved. And so it's just another kind of reminder of that inherent risk. Shelters also have regular turnover of their populations, animals going in and out, not all at the same time, as well as many possible direct and indirect routes of exposure. So in this case, some of the cats who were part of the case definition of this outbreak, they were walking around on a floor where other cats who were also part of the case definition had been vomiting. But there were other cats who were simply touched by a staff member who had touched a roll of register tape that another staff member who was caring for another sick cat 4 weeks earlier had touched. And so the incredible diversity of indirect and direct exposure routes for pathogens that we create through our sort of normal human activities in shelters is something that is really good to kind of keep in the back of our minds when we're dealing with outbreaks and outbreak investigations. As with many, many shelter outbreaks, initially we were kind of flying blind, like we could not identify a cause using commercially available diagnostics. Usually we can. It could be elusive at first, but usually as we go through our diagnostic process, we can. But in this case, we really just had to use our kind of traditional training and look at all the clinical and epidemic data to narrow down what type of pathogen could be causing this and design a sanitation protocol and all of the biosecurity and animal management protocols around that. And this is one place that I usually note that it is important for kind of your basic sanitation and biosecurity protocols to address non-enveloped viruses like parvoviruses, like Fechavirus, and of course our feline panleukopenia and calicivirus, rabbit hemorrhagic disease. All of these non-enveloped viruses that are very durable are only killed by certain disinfectants. So even the basic protocols, not in the face of an outbreak, should address those types of viruses. But really you want, when you're in an outbreak, you always want to review your sanitation protocols as well. We also want to review all animal management protocols in cases like this. So things like, are you in your capacity for care? What's your adoption and transfer driven capacity? What's your length of stay? Is there crowding? Most outbreaks that I've ever investigated as a shelter vet had lots of problems in those areas, but this actually occurred over the winter holidays at a very low capacity time for us, like a low volume time. We didn't find any deficiencies like that. So that was another really interesting thing. Like this could have been so much worse if it had happened in like May or June. We want to encourage shelters and vets to consider novel viruses, not just Fechavirus, which we really need to learn more about, but also just other pathogens that we might not have tests for. If something is behaving strangely, it's really important to kind of believe it. And of course, to exhaustively rule out all of your known stuff, but reach out to academic shelter medicine programs, reach out to your commercial laboratories. People who are doing research need to have clinicians on the ground to partner with, because there is tons of metagenomics research going on around the world, and the mammalian viruses are being discovered at this astonishing clip. But what really needs to happen kind of next for all of these things is to understand their clinical significance or lack of clinical significance in individuals and in populations. So I'd really encourage people to reach out beyond, if something kind of is beyond your expertise and your diagnostic capabilities, as this definitely was for us, reach out for help, because you will find help, and it may end up yielding information that could help even more animals in the future. And I guess the last thing that I kind of wanted to share is that this is Pablo after his adoption, and he's relaxing by the fireplace with his new family. And you'll remember from the previous slide that he's one of the cats who brought this virus to Vancouver. And the management of this outbreak, it required hundreds and hundreds of hours of time from many people in 4 facilities. But I want to kind of, again, bring it back to what we do in shelters caring for populations, and that this is a type of disease that has a high attack rate, so it affects lots of naive animals, and it has a really low mortality, so it affects lots of animals but doesn't kill animals. Diseases like this can become endemic in shelters, and I have worked with shelters that had endemic disease, especially respiratory disease. And I think that when you're dealing with disease, especially new outbreaks of disease, it is so important and so worth it to take every measure and every step that you need to take in order to terminate that disease, because animals that are coming into the shelter should be kept healthy ideally, and you should be able to bring these things to an end. And on the flip side, sometimes outbreaks are dealt with by depopulation. And that's another thing that I think is kind of the opposite extreme from letting something become endemic. But that's another thing that should really be a last resort for us as shelter medicine practitioners. We should be using all the tools in our toolbox to try to bring these outbreaks to an end while learning as much as we can about them. So little Pablo is here to remind us of that. And oh yes, Stephanie.
Wonderful, thank you so much, Emilia. Now back sort of almost at the beginning when you started to talk about the outbreak, Linda had asked, what do you mean by a fecal library?
Oh, yes. So a fecal library was literally just, it sounds so much more glamorous than it actually is, Linda. It's basically just saving all of the feces from diarrheic animals in individual hard-sided closable vials with the date and the animal ID. And then we had the spreadsheet that went with it that told us about the clinical picture of the case. One major learning from this outbreak for us is that your fecal library should include like internal controls. So one of my biggest regrets, that will probably be one of my biggest regrets in my whole life, is not collecting feces from healthy, unaffected cats and not collecting serial samples over time in these cats. We thought it was so remote that anything would even be found, and we were kind of in that survival mode. We had like 75 cats that were sick in 4 facilities, and so I will say that you can always throw the samples away later, but you can never get them back. So your fecal library ideally should have everything labeled, and if you think you might have to do commercial tests, what we were doing initially was we were splitting the samples, refrigerating part, sending that part to IDEX, and freezing the other part immediately, because for future genetic analysis, typically frozen samples that were frozen right away and not thawed and frozen repeatedly are better. That's the library.
Ah, wonderful. Thank you.
Sure.
So Linda also asked, to clarify, is the suspicion that the Fechavirus may not have been in all the samples just because of sample quality?
The short answer to that is no. So I think there's a few possible explanations for this. One is that we don't know that the Fechavirus caused this outbreak. We can't conclude that from the available clinical and scientific evidence. So it's possible that this outbreak was caused by something else. It would have to be some kind of virus that was like so divergent and like so ancient that basically the metagenomics methods didn't pick any of it up in any of the samples. So it is maybe not super likely, but we can't say for sure that this outbreak was caused by Fechavirus. The other thing, so in the human literature, there is work showing that freeze-thaw cycles and like other like sample degradation issues can affect sampling for this type of analysis. So that is definitely possible. The samples that we obtained earlier were of lower laboratory quality than the later ones. But I think that the third possibility is that those of us who are working in shelter medicine know that enteric pathogens in animals are like super fascinating. And one of the things is that there's no enteric pathogen that always causes disease in every single animal that is exposed or infected. And so it's possible that we have this situation where you if the virus could be shed very transiently, as other parvoviruses are. We all know the difficulty with diagnosing canine and feline parvovirus, where it looks and smells and acts like parvovirus, and you like do 4 tests. You like swab the tonsils and you swab the rectum, and you do the tests on all different days, and like they're all negative. And then sometimes the animal doesn't make it. Necropsy, classic parvovirus. That happens actually a lot, especially in cats. With us, we see that all the time. So shedding can be very transient of viruses, and it's most likely in the acute phase of illness. But all the early samples that we obtained, it was like once we realized like, okay, hey, we have an outbreak and we need to collect samples and this is what we need to do with them. And so by the time we were collecting samples from these animals, they mostly just had like diarrhea and were recovering. We weren't getting them in the acute phase of vomiting and diarrhea, except for the last round of samples. So again, if you're building a fecal library, definitely try to get samples in the acute phase, like starting right when animals get sick and then continuing to collect the samples.
That makes sense. So Linda also had another question that said,
Yeah.
What is the BC SPCA's policy about adding singleton kittens to other kitten groups? Because we're trying to balance the risk of them incubating disease, as you found out, versus leaving kittens alone. What is your policy on that?
Yeah, so in general, we don't mix litters of kittens. We generally won't add a singleton to another litter of kittens. But one thing that we do recognize, that I know, again, all of you in shelter medicine and shelter behavior know this, singleton kittens, over their whole lives, there are some behavior risks. So what we will sometimes do is if we have 2 singleton kittens who are both apparently healthy, we will put them together, so we'll pair them. That is, I think, the most common kind of merging of kittens that we do, and it's essentially to it's essentially like weighing the risk and benefit and wanting them to have the lifetime behavior benefit as a cat that's going to be living in someone's home with humans and cats. We don't see a ton, like we don't have a lot of infectious disease transmission in our care, and I think our protocols are pretty robust. So we're very fortunate to be in Canada, where the overpopulation isn't as bad on the West Coast in some areas. And so we generally, like one reason why this outbreak was notable is that it really emerged from the baseline of what we deal with and just came from nowhere. And we were like, oh, this is something. But a lot of the kittens that we see, it's sort of like obvious what's going on with them and whether they're healthy or not. So a lot of it is like individual risk. But yeah, we generally don't add to litters. So I had this slide with the paper. You can grab the link if anybody wants to read it. It's open access, so you can read the full text. This is just a little summary slide of other research that we're doing in animal health here. So we've got a ringworm epidemiology paper coming out in a couple of weeks in the Canadian Veterinary Journal. We're working on characterization of another viral outbreak that happened earlier this year in the winter, and we're using our big ringworm results database to look at doing some other comparisons of different testing methods and trying to look at risk factors for prolonged treatment duration. We also have a partnership with the UBC Animal Welfare Program, and through that we're looking at surrender data as well as a bunch of cat adaptation and cat macro environment type work to really study some areas that haven't been studied, especially in Canada. And I wanted to make sure that I had time to acknowledge everyone that was involved with this work, and not just the paper, but the outbreak itself. First off, I want to say to the cats and the people who experienced this and got through it, and the people who worked so hard to say thank you and acknowledge what they went through. I also want to mention Dr. Karen Van Haften and Mark Takar, our COO of the BC SPCA. They gave literally of their time and funds to make sure that the clinical work and the research analysis that needed to happen here could happen. And without them, this would not have been possible. The paper itself was a collaboration, an international collaboration between a couple of different folks. So Dr. Yanpeng Li at UCSF discovered the novel virus. Mandy and I obviously did the clinical work here at the BC SPCA, and Mandy spent hundreds of hours working with the shelters, going and helping with finding breakdowns in infection control and just support, and also curating all the data. So we had all these individual animal records, like huge binders full, that got distilled down into one simple spreadsheet so we could do all the epidemiology stuff. Neither the outbreak recovery nor the research part, I think, would have been possible without Mandy's contribution. Dr. Ida Altan is at UCSF. Dr. Seguin and Marco Estrada are with IDEX Laboratories, and Jutta Deng and Eric Delwart are at UCSF. I also wanted to mention the contributions of Dr. Roxanne Chan at IDEX I described earlier, Dr. Kate Hurley and Dr. Patricia Pesavento at UC Davis, all of whom really helped us get through the clinical piece, and also helped us with the knowledge and advice to understand what type of sampling we had to do so that these could be useful to researchers someday. And this is my last slide, and there's Grayson with his friend Rocky, and you'll remember that he was there on the first slide shortly after intake. Here he is looking quite relaxed and with his friend, and again, a reminder that these outbreaks that we work on, they involve individual animals, and it's all of our role together to collaborate and to give these animals the best health and welfare that we can while they're with us.
Thank you, Emilia. Alex asked, was there an investigation done at the source shelter after the fact or during the outbreak at all?
Of the Cornell shelter?
Yes.
So like, how did it get into the Cornell shelter?
And did they do any research after the fact? Did they have an outbreak at their shelter, or is it just these 2 patients that came to you and nothing more happened at the source?
Yeah, so that's a great question, and that's actually a question that I've been asked every time I've presented this. So like, how did this happen kind of at the level of patient 0? So the Cornell shelter is one of our shelters. So they were kind of part of all of the investigation and discussion throughout. But when we traced it back to those 3 individual kittens, they came in together as a litter from a rural area, and we weren't able to trace it back any further. Like, they were like there was no population there. It wasn't like a hoarding case. It wasn't like they came from a larger population. They were just like kittens that someone found outdoors and brought into the shelter. So our research collaborators, you know, we were looking at papers on cross-carnivore transmission of parvoviruses in general and wondering whether perhaps it's related to the canine parvovirus, but we just don't know. Sadly, because so much of that outbreak in that Cornell shelter was identified like retroactively, like the outbreak started before it was identified. And by the time it was identified, we were only able to get some very basic samples from a very small number of those cats for like basic fecal analysis, and then they were discarded. So we had no research samples from that shelter at all. And that is another one of my great regrets, is like that we, you know, I think we did our best, but in hindsight, this probably could have been identified in each of the locations where it occurred before it was. And so that's an area that we are focusing on with training and further discussion. And now that this has happened, everyone in our organization is like terrified of any GI illness in any cats, and we get all these like panicked emails if like one cat throws up. So I think we're more aware of it now, just naturally through the process of dealing with this. But yeah, that's a great question, and it's a question that we'll never be able to answer, and I'll always wish that we could have answered it. Sorry, Alex.
That's great. Unfortunate, but we totally understand. Stephanie had asked, if another shelter experiences similar cases, can this test be requested from IDEXX, or what would be the next step?
Yeah, so that is an excellent question, and I don't think we have any of the collaborators on the call. I did let them know about it. So with virus discovery in general, there's lots of viruses being discovered, and kind of the next step with each virus is to try to figure out if it's clinically significant. So the next step with this virus would be looking at either screening like large populations of healthy and sick cats, or like looking for it in specific outbreaks, or doing like surveillance type screening in different shelters and correlating with clinical signs, or doing inoculation studies. These are all things that people are interested in doing and are talking about doing. And then the next steps beyond that, if it turns out to be anything, which, you know, many viruses don't turn out to be anything, would be potentially incorporation into commercial diagnostic panels or tests, and or like vaccine development, those types of things, depending on like the significance of something as a pathogen. So I think a lot more research needs to happen before like anyone will decide that it's worth investing in this as a commercially available diagnostic test. We are in touch with IDEXX. It is worth being in touch with your diagnostic laboratories and asking about this, but because the clinical significance is so the information is so scant, you can't like call IDEXX and request this test at this time. What I will say is that if you use IDEXX, you could call them and say you think you might have this, and they might be willing to put you in touch with the same collaborators that we worked with that have the primers for this virus that are interested in screening more samples. We've actually screened samples from another outbreak and also some healthy cats, just like through kind of opportunistic sampling, working with them, about 20 or 30 cats, and we have not found it again. So we're starting to develop some evidence that it's not like part of a normal healthy cat virome. But you can also reach out to me if you really think that you're having an outbreak of this, not if there's like 1 or 2 vomiting or diarrheic animals, but I would be really interested in talking to you to see if the patterns are similar, and then maybe helping facilitate those connections, because I know that the researchers that we worked with do want to, like, they are open to looking into this more if other shelters have similar outbreaks. Sorry, that was a really long answer. A really short question.
That's wonderful, Emilia. Thank you. You're so detailed. It's great investigation skills. Does anyone else have any other questions for Dr. Gordon?